Hi everyone and welcome to lesson 4 of B3. This lesson is on producing monoclonal antibodies. This lesson is triple only content, so if you're a combined student you do not need to engage with this lesson. As always, you will need a pen and some paper, so if you need to go and get them, please pause the video and get them now. Today's challenge is to describe how monoclonal antibodies are produced, and today's Aspire is to explain how monoclonal antibodies can be used. So the starter for today's lesson is to describe 3 ways in which white blood cells can destroy pathogens and why are antibodies specific. So I'm going to give you 4 minutes to do this starter. Off you go. So the answers that you should have are: white blood cells help to defend against pathogens by, firstly, some white blood cells engulf pathogens and they digest them with enzymes, and this is called phagocytosis. The second one is some white blood cells produce antitoxins which counteract or neutralize the toxins that are released by the pathogens themselves, particularly bacteria. And some of them produce antibodies that can destroy a particular pathogen, and they are specific to one pathogen. So they'll cause the pathogens to clump together so that they can be engulfed. And the next question was, how are antibodies specific? They are specific because they only work on one binding site on one protein antigen, and the shape of the antibody is complementary to the shape of the antigen. So the first thing to talk about with monoclonal antibodies is what's so special about them. So first thing to talk about is monoclonal antibodies are produced from lots of clones of a single white blood cell. And what this means is all the antibodies that are produced are identical and will only target one specific protein antigen. Now this is extremely useful when we're trying to treat cancer, and I'll talk to you more on this later on in the lesson. So the antibodies are produced by a specific white blood cell called a B lymphocyte. And the simple solution would always be to extract B lymphocytes and grow a lot more of these. However, this actually doesn't work very well at all because they don't divide very easily. So what I'd like you to do now for me is, you will know a cell which divides very easily and could very easily fuse with a B lymphocyte. So what do you think that cell could be? So the question is, which cells divide very easily and could fuse with the B lymphocyte? So I'm going to give you 30 seconds to write down your answer. Off you go. So the cells that divide very easily, that hopefully that you thought about and that could fuse with the B lymphocyte, are tumour cells. Okay, so now I'm going to talk you through on how monoclonal antibodies are made. So the first thing that happens is a mouse is injected with a specific antigen, and the mouse's immune system will respond to this. White blood cells then, called B lymphocytes, produce specific antibodies to that antigen. Now we know that tumour cells are able to divide easily, but they can't make antibodies. So what we now do is the B lymphocyte cells are fused with the tumour cells. Now these cells are now called hybridoma cells, and they make specific antibodies. They are screened to make sure they are producing the correct antibody, and then the hybridoma cells are cloned to get lots of identical cells. These cells now all produce the same antibody. These antibodies are now called monoclonal antibodies because they came from a single cloned cell. The antibodies are then harvested and purified, and they're used for a range of purposes. Okay guys, so going with what we've just learned on the previous slide, I would now like you to fill in these blanks for me. Now what I would like you to do is write 1 to 9 on your sheet of paper, and I'm going to give you 3 minutes to come up with the missing words. So 3 minutes, off you go. Okay guys, so now we're going to go through and mark the work. So the first missing word is the word antigen. So it reads, the mouse is injected with a specific antigen and the mouse's immune system responds. Then the second two missing blanks are B lymphocytes and antibodies. So white blood cells called B lymphocytes produce specific antibodies to that antigen. Then the next bit, we know that tumour cells are able to divide easily, which is the missing word, but they cannot make antibodies. So then we actually fuse together the B lymphocytes with the tumour cells. So the word fused and tumour are the words that are missing there. These then are now called hybridoma cells. So hybridoma is the missing word and can make specific antibodies. They are then screened to make sure they are producing the correct ones. And then the hybridoma cells are cloned. So the missing word is cloned to get lots of identical cells. These cells are now all producing the same antibodies. And then the antibodies are then called now monoclonal antibodies because they have come from a single cloned cell. And then the antibodies then harvested and purified and used for a range of purposes. Okay, so now there is a 5-question quick quiz. So I'd like you to write 1 to 5 on your sheet of paper. And here are the questions. I'm going to give you 5 minutes to answer these questions. So what is the mouse injected with and what then happens? Number 2, why can't you just grow more B lymphocytes? Number 3, what is a B lymphocyte fused with and what is the name of this new cell? Number 4, how do you know how to make lots of identical copies of this cell? And once the antibodies are collected and harvested, what happens to them? So I'm going to give you 5 minutes, off you go. So the answer to question number 1 that you should have, so the question was what is the mouse injected with and what then happens. So it's injected with a specific antigen. The mouse's white blood cells then make antibodies which are specific to this antigen. Question number 2, why can't you grow more B lymphocytes? It's because they don't divide very easily. Question number 3, what is a B lymphocyte fused with and what is the name of this new cell? It's a tumour cell and this new cell is called a hybridoma cell. Question number 4, how do you now make lots of identical copies of this cell? You clone the hybridoma cell. And question 5, once all the antibodies are collected and harvested, what happens to them? And they are purified. So I'd like to talk to you now about monoclonal antibodies, specifically in treating cancer. So cancer cells have antigens on their cell membranes and these are called tumour markers. Now, some monoclonal antibodies can bind to the antigens on the cancer cell and it causes them to clump together and then it makes it easier to identify a cancerous tumour, so then you can either treat it or you can remove it. Other monoclonal antibodies can carry toxic drugs or radioactive substances that will bind to and kill cancer cells, so it is really important to help us treat cancer. Now, monoclonal antibodies also have other diagnostic uses. So they can be used in a similar way to cancer treatment and they can help diagnose infections such as HIV and AIDS, herpes and chlamydia. And some monoclonal antibodies have been attached to dyes and they glow fluorescent under UV light, and this can then help disease identification much easier. So another use of monoclonal antibodies is in pregnancy testing. So when a woman becomes pregnant, a hormone is found in her urine called HCG. And pregnancy testing kits that you buy will detect this hormone. So how it works is the bit of stick that a woman will wee on has some antibodies to the HCG hormone stuck to some blue beads. And the test strip has some more antibodies to the hormone stuck onto it. So if a woman is pregnant, the HCG hormone binds to the antibodies on the blue beads. The urine moves up the stick and it carries the hormones and beads with it. And the beads and the hormone then bind to the antibodies on the test strip and the strip that will then turn blue. Okay, so what I would like you to do, guys, now is I'd like you to think about what happens if a woman is not pregnant. What do you think that will happen with this test? So I'm going to give you 1 minute to write down your ideas. Off you go. So what will happen if a woman is not pregnant with this test is the urine will still move up the stick carrying the blue beads, but there'll be nothing to stick the blue beads to the test strip, so the test will not turn blue. So with monoclonal antibodies, there are benefits and limitations. So I would like you to copy down this table, and I would like you to think of any benefits and limitations to using monoclonal antibodies and write them in your table. I'm going to give you 4 minutes to complete this task. Off you go. So the benefits and limitations of monoclonal antibodies are: the benefits can test for pregnancy, can test for diseases such as herpes, chlamydia and HIV, it can treat cancer, and it can be produced very quickly once they've been made for the first time. However, the limitations: some monoclonal antibodies have caused unwanted side effects, they are very expensive to produce, and there are some ethical issues, how some people disagree with using mice to produce monoclonal antibodies. So guys, we are now at the summary at the end of the lesson. As always, I would like you to copy down these summary notes. I will give you 5 minutes to do that. If you need any more time, as always, please pause the video. So today's summary: white blood cells produce antibodies which attach to specific antigens on pathogens. White blood cells which produce antibodies are called B lymphocytes. Monoclonal antibodies are produced from lots of clones of single white blood cells. This means that all antibodies produced are identical and will only target one specific protein antigen. A mouse is injected with an antigen and the mouse's B lymphocyte cells will produce antibodies. Antibodies are combined with tumour cells to make hybridoma cells. Hybridoma cells are then cloned and the antibodies are then harvested and purified. And monoclonal antibodies are used to treat cancer, detect diseases such as HIV and herpes, and also are used in pregnancy tests. So I'm going to give you 5 minutes to complete these summary notes. Off you go.